In 2008, deep inside a limestone cave in the Altai Mountains of southern Siberia, Russian archaeologists pulled a tiny fragment of bone out of the dirt, a sliver from the finger of a child who had died there tens of thousands of years earlier. It was so small and so unremarkable that it easily could have been catalogued, boxed, and forgotten alongside thousands of other fragments already sitting in storage. Instead, that fragment ended up on a lab bench at the Max Planck Institute for Evolutionary Anthropology in Germany in the hands of a geneticist named Svante Pääbo, and by 2010, the DNA extracted from it had done something no fossil discovery had managed before. It didn't match Neanderthal DNA. It didn't match modern human DNA. It belonged to something else entirely, a population of ancient humans nobody had ever known existed, hiding in plain sight inside a cave that archaeologists had already been digging through since the 1970s without realizing exactly what they'd stumbled onto. The finding was significant enough that the journal Nature carried it, and the U.S. National Science Foundation, which had partially funded the underlying research, issued its own public announcement calling it evidence of a distinct group of archaic humans existing outside Africa more than 30,000 years ago, at a time when Neanderthals were thought to have dominated the whole of Europe and Asia, largely unchallenged by any other archaic population. Genetic sequencing of the finger bone, which had belonged to a girl somewhere between 5 and 10 years old at the time of her death, showed she was neither Neanderthal nor modern human, but shared an ancient common origin with Neanderthals while having followed a dramatically different evolutionary path ever since. It was, by any measure, an extraordinary claim to make from a single, tiny bone fragment, and it demanded a second independent line of evidence before the wider scientific community would take it entirely seriously. The cave was called Denisova Cave, named after a hermit who was said to have lived there centuries earlier, and the population identified from that finger bone would come to be known as the Denisovans, the first entirely new branch of the human family tree discovered through genetics alone, without a single complete skeleton, without a skull, without so much as a jawbone to look at. Just a bone fragment smaller than a coin and a handful of genetic letters that didn't line up with anything else on record. It remains, even now, one of the strangest and most quietly dramatic discoveries in the entire history of human origins research, because everything scientists have learned about this third species of human has come almost entirely backward, from genes first and physical appearance only much, much later. To understand exactly how startling that finger bone discovery was, you have to understand what scientists thought they already knew about human evolution in Asia at the time. The prevailing assumption held that after modern humans left Africa roughly 60,000 years ago and began spreading into Europe and Asia, they encountered essentially one other kind of ancient human already living there, the Neanderthals, well known from over a century of fossil discoveries across Europe and the Near East. Asia itself, and Siberia in particular, was considered comparatively poorly understood territory, a vast region where the fossil record was thin and where nobody expected any major surprises. The Denisovan finger bone blew that assumption apart in a single stroke. Here was proof that an entirely separate lineage of ancient humans, distinct from both Neanderthals and modern humans, had been living in the heart of Asia the whole time, and nobody had known to look for it because nobody had any physical evidence substantial enough to suggest it existed. The genetic analysis itself, once it came back, left very little room for doubt. Sequencing showed that the girl whose finger bone had been recovered shared an ancient common origin with Neanderthals, but had followed a dramatically different evolutionary path since that split occurred. Genetic comparisons eventually placed that split the moment the ancestors of Denisovans and the ancestors of Neanderthals diverged from each other as separate populations. Somewhere in the range of 400,000 years ago, an enormous stretch of separate evolutionary history condensed into a single fragment of finger bone smaller than a fingernail. A tooth recovered from the same cave complemented the genetic story perfectly. Paleoanthropologist Bense Viola, who studied it at the time, called the tooth amazing precisely because its shape fell completely outside the normal range of variation seen in either Neanderthal or modern human teeth, giving researchers physical structural confirmation to sit alongside the purely molecular evidence, two independent lines of proof pointing at the same startling conclusion. This was neither of the two human lineages science already knew about. What makes Denisova Cave itself such an extraordinary sight isn't simply that Denisovan remains turned up there. It's that the cave, when researchers eventually dug deep enough into its sediment layers and applied increasingly sophisticated genetic techniques, turned out to have hosted not one, not two, but 3 separate human species across its long occupation history, making it one of the only known locations on Earth where Denisovans, Neanderthals, and modern humans can all be directly documented living within the same physical space, even if not always at exactly the same moment. A landmark study analyzing ancient mitochondrial DNA recovered directly from the cave's sediment without needing a single visible bone fragment to work from, reconstructed a rough timeline of who occupied the cave and when. Denisovans arrived first, with genetic traces appearing in sediment layers dated to roughly 250,000 years ago, tied to early Middle Paleolithic stone tools. Neanderthals showed up later, their genetic signature first appearing around 170,000 years ago. The two populations appear to have used the cave on and off, likely at different times rather than constantly side by side, for well over 100,000 years, right up until roughly 45,000 years ago, when the genetic evidence shows modern humans finally arriving on the scene as well. That kind of layered, multi-species occupation produced some genuinely startling individual discoveries along the way. In 2010, in a section of the cave known as the East Gallery, researchers found a Neanderthal toe bone, identified through DNA analysis, sitting in a layer that dated to roughly the same period as the original Denisovan finger bone, meaning that at some point both populations had left physical remains in strikingly close proximity within the cave's timeline. Neanderthals also left behind their own distinctive stone tools throughout the site and surrounding region, a style archaeologists call Mousterian technology, mostly dating to around 40,000 years ago, while other layers held more sophisticated stone tools and bone artifacts that likely belonged to modern humans instead. Untangling exactly which layer belonged to which species, and whether any of these populations ever crossed paths inside the cave at the exact same moment, rather than simply using the same shelter across different centuries, remains a genuinely difficult puzzle, one complicated by the fact that cave sediments shift, mix, and compress over tens of thousands of years in ways that can blur even careful excavation work. But the most direct, unambiguous proof that these different populations didn't just occasionally share the same physical space, but actually intermingled on the most intimate level possible, came from a single, tiny bone fragment discovered years later and analyzed in 2018. The fragment itself was almost comically unremarkable to look at. A splinter of a bone roughly 2 centimeters long, initially cataloged among thousands of other unidentified pieces of debris pulled from the cave floor over decades of excavation. The exact kind of fragment that could easily have been tossed aside as unremarkable animal bone if nobody had bothered to run it through genetic testing. Researchers extracted DNA from it and found something that had never been documented anywhere else in the human fossil record. This wasn't simply another Denisovan or another Neanderthal. It was the direct offspring of one parent from each group, a girl who died as a young teenager with a Neanderthal mother and a Denisovan father. Scientists nicknamed her Denny. Her genome offered something no amount of comparing separate populations from a distance could ever fully prove. Physical, undeniable evidence that Denisovans and Neanderthals didn't just occasionally interact, they had children together, real families that produced real offspring, in what appears to have been a genuinely ordinary, unremarkable event. From the perspective of the people involved at the time, however extraordinary it looks to us discovering it nearly 90,000 years later. The odds of stumbling onto a first-generation hybrid individual, rather than someone whose mixed ancestry was diluted across many prior generations, are themselves worth pausing on. Given how few Denisovan and Neanderthal remains exist anywhere in the world, finding a single bone fragment that happened to belong to a direct, one-generation product of exactly this kind of pairing suggested that this sort of mixing may have been considerably more common at the time than the sheer rarity of the fossil record alone would ever have hinted at. Even more strikingly, subsequent genetic analysis suggested that this kind of mixing wasn't a one-off accident either. Denny's own father, the Denisovan side of her ancestry, carried genetic traces suggesting that some of his own more distant ancestors had themselves included Neanderthals, indicating that interbreeding between these two populations happened repeatedly across generations, not as a single freak encounter, but as an ongoing recurring feature of life in this region for tens of thousands of years. In other words, the discovery of one single hybrid individual didn't just prove that interbreeding was biologically possible between these two lineages, it hinted at a much longer, quieter history of two supposedly separate species repeatedly choosing, or simply happening, to build families together across an enormous span of time in the same shared landscape, likely within the very same cave system where their remains would eventually be found lying almost side by side. The story of the Denisovans doesn't stay confined to that one cave in Siberia either, and this is where the geography of the discovery becomes almost as surprising as the genetics themselves. In 2019, researchers studying a mandible, a lower jawbone that had been recovered decades earlier from Baishia Karst Cave, a limestone cave sitting more than 10,000 feet above sea level on the northeastern edge of the Tibetan Plateau, used ancient protein analysis to tentatively link it to the Denisovan lineage as well, a fossil that came to be known as the Ziahi mandible, dated to roughly 160,000 years old. The jawbone itself had actually been found by a local monk decades earlier, and later donated to a research institution sitting unidentified for years before modern analytical techniques were able to place it within the Denisovan lineage at all. Another example of a genuinely significant fossil hiding in plain sight simply because nobody yet had the tools necessary to recognize what they were looking at. Later work went even further, recovering actual Denisovan mitochondrial DNA directly from the cave's sediment itself, dated to somewhere between 100,000 and 60,000 years ago, with some evidence hinting the population may have persisted there as recently as 45,000 years ago, a date that would place Denisovans in the same region and same rough time frame as early modern humans beginning to arrive in Northeast Asia. One researcher involved in analyzing the Tibetan cave site put the scale of the ongoing discovery in blunt terms, noting that after roughly a decade of work, the site had proven far richer than anyone on the team had originally expected, yielding hundreds of stone tools, animal remains, and ancient DNA from just a handful of small excavation units, with the clear implication that a great deal more waits to be uncovered there still. Researchers have also noted that fossilized animal bones recovered from the same high-altitude Tibetan site appear to have migrated into the region from as far away as the foothills of the Himalayas to the southeast, hinting that whatever drew large mammal populations up onto the plateau in the first place may have been exactly what drew Denisovan communities to follow them there as well, adapting to one of the harshest environments on the planet in the process. The geographic spread matters enormously for understanding just how successful and adaptable the Denisovans actually were as a population, something the fossil record alone, with its handful of scattered fragments, could never fully demonstrate on its own. A population that could survive both the cold mountain forests of southern Siberia and the extreme high-altitude environment of the Tibetan Plateau, sitting more than 3,000 meters above sea level with dramatically reduced oxygen levels, was clearly not some narrow, geographically limited group clinging to survival in a single valley. And that adaptability left behind a legacy that didn't disappear when the Denisovans themselves eventually did. Genetic studies of modern human populations have found that people living in Melanesia today, particularly in Papua New Guinea, carry as much as 4 to 6% Denisovan DNA in their genomes, among the highest levels of archaic human ancestry found anywhere in the world, evidence of interbreeding between Denisovans and the ancestors of modern humans that most research now dates to somewhere around 46,000 years ago, with some more contested evidence suggesting additional separate interbreeding events may have occurred as recently as 30,000 or even 15,000 years ago. Even more strikingly, modern Tibetan populations carry a specific Denisovan-derived gene variant called EPAS1 that helps regulate how the body responds to low oxygen levels at high altitude. A genetic gift, essentially, passed down from a species that had already spent tens of thousands of years adapting to exactly that kind of extreme environment, now quietly helping modern humans breathe more easily on the same mountain plateau their distant Denisovan ancestors once called home. Researchers have described the EPAS1 variant as one of the clearest documented examples anywhere in human genetics of a beneficial trait being inherited wholesale from an archaic, now extinct population, rather than evolving independently within modern humans themselves, meaning that millions of people living at high altitude today are, in a very real physiological sense, still benefiting from evolutionary experimentation that Denisovans had already run to completion tens of thousands of years before any modern human ever set foot on the Tibetan Plateau. For all of that genetic richness, though, one enormous gap sat at the center of Denisovan research for essentially the entire 15 years following that first fingerbone discovery. Nobody actually knew what a Denisovan looked like. Every single piece of evidence recovered up to that point, the fingerbone, the tooth, the Tibetan jawbone, Denny's tiny fragment, was simply too small and too incomplete to reconstruct anything resembling a full face or skull. Scientists could describe, in exhaustive genetic detail, how Denisovans related to Neanderthals and modern humans, which genes they'd passed down to living populations, and roughly where and when they had lived, while remaining almost completely unable to say what one of these people would actually have looked like standing in front of you. That finally changed in June 2025, and the way it changed involves one of the more remarkable detective stories in the entire field of paleoanthropology. Back in 1933, in the city of Harbin in northeastern China, a laborer working on bridge construction during a period of Japanese occupation uncovered a massive, nearly complete human skull. Fearing the fossil might be confiscated by occupying forces, the man secretly hid it at the bottom of a well, where it remained completely unknown to science for more than 80 years. The skull only resurfaced decades later, and when researchers finally examined it properly, its enormous, robust features, a brain case measuring around 1,420 cubic centimeters, square eye sockets, and a massive jutting brow ridge, looked unlike anything definitively matched to an existing human lineage. In 2021, one team of researchers formally described it as an entirely new species altogether, giving it the name Homo longi, nicknaming the specimen Dragon Man after the region where it had been found. For a few years, that classification stuck, and the aging skull, dated to at least 146,000 years old, sat awkwardly in the scientific literature as a supposedly distinct fourth branch of the human family tree, one whose actual relationship to Denisovans, if any, remained pure speculation based only on its general age and rough geographic proximity to other suspected Denisovan sites. The Dragon Man label didn't last. Researchers led by Qiaomei Fu at the Institute of Vertebrate Paleontology and Paleoanthropology in China, working alongside colleagues including Qiang Ji, went back to the nearly 100-year-old skull with tools that simply hadn't existed in 1933 or even in 2021. The ability to extract genetic material and ancient proteins from dental calculus, the hardened plaque built up along a fossil's gum line over a lifetime, sealed and preserved for 146,000 years. The team managed to scrape out a mere 0.3 of a milligram of that ancient calculus, and although nearly 4/5 of the human genetic material recovered turned out to be modern contamination from decades of handling, likely introduced by researchers, museum staff, or simply the conditions the skull had been stored in over the decades. The small remaining fraction of authentic ancient DNA told an unmistakable story. It was mitochondrial DNA, the kind passed down exclusively through the maternal line, and it matched, closely and consistently, mitochondrial DNA already sequenced from confirmed Denisovan individuals recovered from Siberia's Denisova cave, roughly 2,500 miles away. In a parallel, independent study, the same research team analyzed ancient proteins preserved in the skull's petrous bone, one of the densest bones in the human body and famously good at preserving biological material, over enormous stretches of time, comparing the resulting protein sequences against modern humans, Neanderthals, Denisovans, and non-human primates alike. The proteins pointed to the exact same conclusion the DNA had already suggested. This wasn't some undiscovered fourth species. It was a Denisovan, and not just any Denisovan, but the single most complete skull ever recovered from this entire lineage, finally giving scientists something they'd been missing for a decade and a half: an actual face to attach to the genetics. Fu herself, describing the significance of having two independent molecular methods, ancient DNA and ancient proteins, both converge on the same identification, noted that together the two studies dramatically strengthened the case for Harbin representing a genuine Denisovan individual, a rare instance in ancient genetics of two entirely separate analytical techniques cross-checking each other's conclusions on a single, singularly important fossil. Outside experts who reviewed the findings without having been involved in the research themselves described the molecular evidence as compelling. Chris Stringer, a veteran human origins researcher at London's Natural History Museum who has spent years collaborating with Chinese scientists on separate morphological analyses of the same skull, called the new genetic and protein data potentially very important, noting that combined with his own team's work, the case for Harbin representing the most complete Denisovan fossil found anywhere to date had become increasingly strong. Frida Welker, a biomolecular paleoanthropologist at the University of Copenhagen, who has recovered Denisovan proteins from other candidate fossils but wasn't part of this particular study, was similarly direct, describing the protein signature as pointing to Denisovan ancestry, with any other explanation being very unlikely. For a scientific mystery that had sat unresolved for 15 years, robbing an entire species of any recognizable face, it was about as clean and dramatic a resolution as ancient DNA research gets. The story didn't stop there either. Barely 3 months after the Harbin skull's Denisovan identity was confirmed, a separate study published in September 2025 proposed extending the same lineage even further back in time and across an even wider stretch of territory, suggesting that human remains recovered from 6 additional sites across China might belong to the same broader Homo longi group now genetically tied to the Denisovans, including remains from Yunxian, dated to an almost unbelievable 1 million years old, a figure that, if the connection holds up under further scrutiny, would push the deep roots of this lineage back to a point in time that dwarfs even the 400,000-year split from Neanderthals that originally defined the group. If that connection is eventually confirmed, it would mean the Denisovan story doesn't just stretch across 400,000 years of separation from Neanderthals, but potentially reaches back to a population that was already establishing itself in East Asia, at a point in time when even Homo erectus was still a relatively recent arrival on the world stage, an almost dizzying expansion of a story that started with nothing more than a single finger bone barely 15 years ago. Other researchers, studying this same expanding cluster of fossils, have proposed folding still more specimens into the picture, including the Tibetan Xiahe mandible and the Penghu jawbone recovered from waters off Taiwan, sometimes under yet another proposed species name, Homo juluensis. reflecting just how unsettled and rapidly evolving the taxonomy around this entire group remains, even after the genetic confirmation at Harbin. Scientists are, in effect, still actively arguing over exactly how many previously separate-looking fossils scattered across China actually belong to one single, geographically sprawling Denisovan population, and how far back into the deep past that population's roots genuinely extend, a debate that will likely take years, and quite possibly several more genuinely surprising fossil re-identifications, to fully settle. Stand back and look at the full arc of this story, and what you're left with is one of the strangest sequences of discovery in the entire history of paleoanthropology. It began with a fragment of finger bone so small it could have been mistaken for animal debris, sequenced not because anyone expected to find a new species, but because a careful geneticist decided it was worth checking anyway. It grew through a single tiny bone fragment belonging to a teenage girl who turned out to be living proof that two entirely different kinds of humans had fallen in love, or at least reproduced, deep in a Siberian cave nearly 90,000 years ago. It expanded across an entire continent, from frozen mountain caves in Siberia to oxygen-starved plateaus in Tibet to a jawbone recovered from the seafloor near Taiwan. And it finally arrived, 15 years after that first finger bone was sequenced, at a face, a massive, heavy-browed skull that had spent 80 years hidden at the bottom of a well, specifically to keep it out of the wrong hands, only to resurface decades later and answer, almost by accident, the single question that had haunted an entire branch of human evolutionary science since the day the very first Denisovan bone was ever identified. What makes this particular story so unusual, even among the frequently strange history of human origins research, is the order in which everything unfolded. Normally, a new human species starts with bones, a skull, a jaw, something a person can physically hold and study, and only much later, if the discovery is exceptionally lucky and exceptionally well preserved, do genetic and molecular details get layered on top of that physical foundation. The Denisovans arrived in exactly the opposite order. Science understood their genetics, their approximate age, their evolutionary relationships to Neanderthals and modern humans, and even their precise genetic contribution to living populations across the Pacific and the Himalayas, all of it, years before anyone could point to a single face and say with confidence, this is what one of them actually looked like. Somewhere out there, in museum drawers and half-catalogued fossil collections scattered across Asia, there are almost certainly more Denisovans waiting to be recognized, hiding in exactly the same way that fingerbone once did, indistinguishable from ordinary debris until someone with the right tools finally thinks to look closely enough